Hi everyone, it's Mr. Sinti, and I'd like to talk to you today about Mendel's experiments. And his experiments have to do with determining patterns of heredity, and not just in pea plants, but in all living things. Now, I'm not saying that Mendel has discovered everything in genetics, but in the 1800s he was, clearly ahead of his day, and he gave us a lot of patterns and a lot of potential for discovering the things that we know today about how genetics works. And so let's dive into that conversation. What I want to say about Mendel's experiments is that he started, if you may recall this from a previous video, he started with true breeding plants. And what I mean by true breeding plants is that he looked at a particular characteristic, like for example purple, and by self-pollinating it, in other words by taking pollen from the anther and putting it on the stigma of the same plant, and a pod would form and the seeds would be born and planted, the plants would always be purple. So purple, 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 purple. So he was pretty confident that there was a pure purple plant. But then what he did, if you recall this, is that he crossed his pure purple with pure white, and all the plants of that cross were purple. So in the first generation, or F1, he called these plants hybrids. And so let me just sort of write that there. Let's say that these are the first generation plants, and we're going to call them the F1, and we're going to call them hybrids right there. And so what's interesting about the hybrid is that he suspected that the hybrid was part purple and part white. And so what's fascinating about that is that he was absolutely correct about it. And so when we now know today, because we have microscopes and we know a lot more about chromosomes, back in Mendel's day in the 1800s, they were completely unaware of this. And so we now know that a pea plant, for example, let me just draw it over here. I won't draw the plant literally, but I'll just say like a cell of a hybrid pea plant has a chromosome from one parent and a chromosome from the other, and these are homologous chromosomes. And so if we say that there's a particular gene for petal color, let's go here, let's say the location is right here for petal color. We could say that it's purple like this, because these are the same sister chromatids, and let's go little p for white. This is what a pea plant would look like that's purple but was a hybrid, meaning that it had both purple and white genes. And so you can see here, now this only has one piece, the other piece is not drawn in. I could draw the other sister chromatid, but this person, or this particular plant, is hybrid. It has purple and it has white. And when you put both of those together, Mendel wrote in his documents that one allele dominates over the other one. And so this is capital and this is lowercase. And so I just wanted to establish that. So Mendel developed a hypothesis to explain a lot of related results that he was getting to. And one of them was that there was contrasting factors. He didn't use the word allele, but there were contrasting factors like purple and white. And it seemed to him that plants had two factors for each particular trait, and that's the truth. In a pure plant, it was purple, purple, and a pure white would be little p, or white, white, and then in a hybrid would be purple and white. And so when you take this a little bit further, you could say that each diploid organism inherits one from the other, and so each of those get segregated into the gametes. So if I may, I'm going to take a moment and sort of illustrate that over here. Let's take a look. If I said that I had a pea plant right here, let's say that it's a pea plant that looks like this, and it has a purple on one chromosome and it has a white on the other one. I'll go like this. And so what we now know today about meiosis turns Mendel's discussion into something that really makes sense. Like, for example, we know that a somatic cell or a body cell or a pea plant cell is diploid. So they have a maternal and a paternal homologous pair. And so in order to make pollen, so this is a, picture this on the side. I'll do a visual. So here's a pea plant like this that has purple, I know it's blue, but purple petals. And so this is what it looks like, but inside this particular plant, if this is a hybrid, it has both a purple and a white combination of genes. And so in order to make pollen, so this is what we're talking about, in order to make pollen in the anther, now that requires meiosis. And so let's walk through that. This particular hybrid purple plant makes two kinds of pollen. And you're like, really? How's that? How can it make two kinds of pollen? I mean, you may know pollen is the sort of looks like this under the microscope, but basically it's a yellow powder, and it's the gamete. And it's like, well, how can it have two kinds of it? It's purple. What else would it be? Well, when you're making meiosis, what happens is meiosis 1 will separate the homologous pairs, so that's purple and purple, and then this one over here, the other chromosome goes over here, so that's white and white. And then when these divide in meiosis 2, like this, what do you get? This is the pollen, or if you will, the gametes. And what's happening is this would be white, white, and this would be over here, of course, purple and purple. Now I know there's 4, but there's only 2 kinds. So this purple hybrid plant is literally making 2 kinds of pollen. And Mendel knew that. I think that's incredible. He didn't know chromosomes, he didn't know alleles, and he didn't know meiosis 1 and 2. But what he did say about this was this. He said that in the hybrid, if there's a purple factor and a white factor, what he said is that the 2 factors segregated in pollen formation, that they segregated. That's exactly what they do. And that occurs during anaphase 1 of meiosis. But he was right about that. I found that that's very impressive. So just want to take that little sidebar right there. And so what's further fascinating is that when you consider the possibilities, so at each location in the pea plant, and the true breeding pea plant, the plant had PP, in other words, purple, purple, and the white one had little p for the white. And so the hybrid purple one was big P, little p. And so what's interesting is when you look at this possibility, and this is a great diagram, his first cross was pure purple. When I say pure purple or true breeding, I mean that it's both capital P, that's a diploid. And then when it makes the pollen, the only kind of pollen you can get from this is capital P. And then white is little p. So when you put the pollen and the egg together, you make a diploid plant that's purple. So big P, little p. So this is what I was just going over. What's interesting is this purple plant makes 2 kinds of pollens, as I just said, a big P and a little p. And so when you look at the possibilities, let me do it over on the side here. When you look at the possibilities from this plant is producing a purple pollen and a white pollen. Okay? But then it has to meet up with the egg. And so it's either the same plant, self-pollination, or cross-pollination. If you cross it, now to show the possibilities, you can make a box like this. It sort of looks like a cross, and at the head or the top of each column are the gametes. So this is the pollen purple, this is the pollen white, and then if you cross it with another hybrid purple, you can do purple over here, and little p is white. And then when they come together, let me use another color for emphasis. When they come together like this, in other words, if that pollen meets up with that egg, it's going to be big P, little p, or purple. And then if these two come together, right here, and this one, it's going to be also purple. And this one is also purple. But when that pollen meets up with that egg, white reappears. So in the F2, in the second generation, Mendel saw 25% of his plants coming back white. Now, that's kind of mind-blowing because most people at the time thought the blending idea was happening. If you mix purple and white, maybe you get like light purple. And then light purple and light purple make light purple. But no, white comes back. And it's like, well, I don't know, maybe this white is different than, no, that white is as pure white as its grandparent. And what's interesting, and you're like, well, what about these purple ones? These three purple ones, some of them are pure purple, and some of them are hybrid purple. But there's certainly three of them. So 75%, 25%, or 3 to 1. So it's totally fascinating. And so what I want to say about that is if you cross, like say this plant right here, you're just looking at it, and you're like, oh yeah, okay. So if I ask you what is the genetic makeup of this purple flower, you shouldn't be able to tell me, because just by looking at it, you don't know if it's capital P, or capital P, lowercase p, do you? You don't know. And like this one, when you look at it, you can say, well, it's white. I think it's little p, and you would be correct. Well, what would happen if you crossed this purple and this white like this, and we made that Punnett square? And let's say that we were crossing it like this. We make the Punnett square. And then, of course, this is the diploid. This is the body cell. But so to make pollen, we would do this, meiosis. This represents the pollen, little p, little p. Let's say that this plant, we didn't know it at the time, but let's just say that it was this one. Say it was hybrid. And so this is the body cell. So it'll make two kinds of pollen, a purple pollen and a white pollen. As it turns out, what you would get from this is purple, white, and white. You'd get 1-to-1 ratio, or 50%, 50%. The first white flower that you get from this cross will immediately tell you that the plant had to be a hybrid. If all the plants were purple, in other words, big P, little p, you'd expect it to be pure. And so that's kind of a cool thing. And so when you look at these pea plants, like Mendel was doing, he started writing in his journal that one factor seemed to suppress the other or dominate. And so he referred to this as the dominant factor, and that the other characteristic, the other factor, was recessive. And so we now call these dominant alleles and recessive alleles. So purple seems to dominate over the white. And so I just wanted to point that out. And this is a picture. Remember I was holding up this little massage ball a little bit. This is really like what pollen looks like under the scanning electron micrograph. And so what I'm trying to get across to you is that a pea plant, here's a leaf like this, and here's a leaf, and in the flower like this, up here in the anther is where the pollen, this is the pollen right here, that's coming off the anther. This is what it looks like under high magnification. So if this is a hybrid purple, this is hybrid purple, that means that some of these pollens have purple in them and some of them have white in them, 50%. Isn't that weird? Even though the plant is purple, it has two different kinds. And so it depends, like which pollen is going to fertilize the egg. And so over here in the pistil, right here, you got an egg. And so, not drawn to scale, but the egg you're going to have different kinds of eggs because these segregate. So say you had a little p, and then this pollen was little p. If those two come together, it's going to make a pod, and the seeds are going to be white as a result of that. Little p makes white. And so I find that to be very interesting. And so Mendel called this his law of segregation, which we now know today as being meiosis, and how the homologous chromosomes segregate in gamete formation. And so some useful vocabulary that I've been reluctant to use, but now I'm going to officially bring it out, is that when the alleles are the same, let me just draw that out over here. When we have a chromosome that looks like this and a chromosome that looks like this, homologous pairs, and both of them are, say for example, we're looking at height, both of them are big T for tall, we could say that this plant is homozygous. Or we could just simply summarize this as saying big T, big T. And when it produces pollen, it can only produce tall pollen. What else can you do? But if a plant is big T, little t, we call this heterozygous when the alleles are different. And so when this plant produces pollen, it produces big T, little t. And so we have this term homo meaning the same and hetero meaning different. And it's a zygote because it comes to find out that when you're doing like a big T pollen is getting together with a little t egg, the result is the zygote, or zygote. Yeah, you know where this is going, zygote. Big T, little t, so these two are different. And what the plant looks like, its physical characteristics, it's purple, it's white, it's tall, it's short, it's green, it's yellow, it's free lobes, it's attached lobes. The physical outcome of the physical appearance of the organism, the physical manifestation of the gene is known as the phenotype. And the genetic makeup of the organism is known as genotype. So when I'm saying a purple flower, I'm talking about phenotype. So what does it look like, and then what is the genetics of it? So big T is genotype. Big T, little t is genotype. Little t is genotype. These two are tall. That's phenotype. And this one right here is short. That's phenotype. So those are very useful terms in genetics. This is what I was kind of smiling about before. You might recognize this. These are not heterozygous, but heterozygotes. And so when you're looking at Mendel's F2 cross, you can see he got this 3-to-1 ratio right here. And so when I say 3-to-1, what I really mean to say, and what I could have been saying before, is what's the phenotypic ratio of the offspring? In other words, what do the phenotypes look like? 3 purples to 1 white is phenotype, the physical appearance. If I said what's the genotypic frequency, let me do the cross and to refresh you. Like for example, let me go green there. If I do right here a hybrid purple crossing with a hybrid purple, what I get is homozygous purple, I get heterozygous purple, I get a heterozygous purple, and a homozygous white. And so, as you can see here, I get 1 homozygous dominant, 2 heterozygous, right here, this is the 2, and the 1 homozygous recessive. So the genotypic ratio is 1 to 2 to 1. 1 to 2 to 1. Whereas phenotypically, what we're dealing with here is 3 purples to 1 white. And that'd be that. And so that's also very useful. So what's curious, and I went over this example with you before, if you're looking at a plant that's exhibiting a dominant phenotype, in other words, if you're just looking at a purple flower, you don't know its genotype. You don't know if it's homozygous dominant or heterozygous. So in order to do that, you would perform what's known in genetics as a test cross. And let me just write that out. A test cross And the reason it's called a test cross is because you're trying to test out to see. You don't know. You don't know if it's homo or hetero. And so you just look at the P's in the next generation. If they're all purple, you suspect that it's probably homozygous. If it's 50-50, you suspect that this is heterozygous. And you're like, well, why is this the question, not this? Because something showing a recessive phenotype has to be homozygous recessive. So I hope that was clear. And that's known as a test cross, where you're trying to figure out something. I like this example. We talk a lot about pea plants because that's what Mendel worked with, but I'm not sure if we're altogether that interested. Like, dogs are very interesting. Most people love dogs, maybe a little bit more than pea plants. As it turns out, if you got this yellow dog over here, or this blonde-colored dog, it's a female, and we know from previous crosses that this fur color happens to be recessive. So she's homozygous recessive. We know this. Just by looking at the dog and know that it's blonde color, we know it's, say, for example, little e, little e. But this sort of dark furred dog over here, this black dog, I don't know. Like, it could be big E, little e, or it could be big E, big E. Let me just write that in just to make sure. So this one we know is this. This one we do not know if it's this or this. Can you tell just by looking at the phenotype? You're like, well, it says it right here. Well, we don't know that. So when you look at the sort of an unknown grab bag here, when you look at if these have children, these two dogs have children, and all the dogs were black, we would suspect this. If some were black and some were blonde, or blonde fur, then we'd suspect that the dog was big E, little e. And it looks like that's the case. So this is a good example of a test cross. But I do want to point something out to you that's very important. That Gregor Mendel, though he contributed so much to our ideas of genetics and heredity, it was many, many years ago. And Mendel had the good fortune, to be choosing traits in a pea plant that only had one gene corresponding to a big phenotype, like tall and short, round and wrinkled, purple and white, and same with these dogs. That most often there's many genes involved in a phenotype, and I will come back to that, but I just wanted to point it out. I don't want to give you the impression that there's just one gene for a phenotype. It's often there's more genes, in what we call polygenic. And so Mendel thought that these alleles separate, and that it was his law of segregation. And so one of the things that I want to talk about now is what happens if you have two traits. Now, what happens there? Well, when he considered two traits at a time, and you're like, what do you mean by two traits? Well, you could have a plant that's tall, that's a characteristic in a pea plant, and that it has purple petals. So that's a separate trait. And you can say that, oh, all right, so if it's tall, it's either this or it's this. And if it's purple, you might know that it's either this or this. So it could be heterozygous or homozygous. And that's two different traits. And so what Mendel was interested in doing was considering two-trait crosses. And when he did these two-trait crosses, he saw patterns there as well. And so let's talk about these patterns. He was, when he considered a single hybrid cross, it's considered to be mono. That's what we've been looking at so far. And so when you do two traits, it's considered to be di for two traits at the same time. So if the plant is dihybrid, that means that it would be like, for example, ooh, it's really big. Calm that down, sorry. If you're doing a dihybrid, like, it would be big T, little t. Now remember, hybrid was a term that we were using before. We've since replaced it with heterozygous. So here's a heterozygous or a hybrid. So it's tall, short. The phenotype would be tall. And let's say purple and white. So this would be a tall purple plant. And it's hybrid for both. So this would be a dihybrid. The interesting thing about it is if you saw a tall purple plant, you wouldn't know if it was homozygous tall or homozygous purple. You wouldn't know that. But let's just say that you were doing this. And so let me set up this cross for you. What would you say if you had, let's go over here, what would you say if you had a plant that was big T, little t, and big P, little p? You're like, okay, let's consider, like, what are the possible gametes from this? And so the possible gametes for this are, when these segregate and you form pollen like this, what can happen is you can have a big T go over to this and a big P go over to this one. So that's one possibility. And then over here you can have a big T and a little p go into this one. That's a second possibility. But then there's some other possibilities as well. You can have a little t and a big P, and a little t and a little p. So how do you like this? This dihybrid plant can actually produce 4 different kinds of pollens for just these 2 traits. Now I'm not even considering all the traits, just 2 at a time. So a dihybrid can make 4 different kinds of pollen. And when you cross that with, let's just say, another dihybrid over here, this plant can also produce 4 different kinds of pollen. And so if you're considering 4 different pollens from 2 different plants, your Punnett square gets kind of large. Like, for example, it's 4 times 4. So what you can do is something like this. You can do big T, big P, big T, little t, big P, and little t, little p. Those are the 4 possibilities. Remember, these are the gametes. This is the pollen. This is the egg. And when they come together, it represents fertilization. So you're like, well, this is crazy. Let's just do a couple of them and get a sense of this. Like, if that pollen fertilized that egg, what you would get is big T coming from over here, little t coming from here, big P, and little p. Phenotypically, what this would look like is a tall and purple. And so when you do this cross, it turns out that there's 9, 3, 3, 1. There's 16. That adds up to 16. 9 is tall and purple, meaning like the dominant. The 3 is dominant recessive, meaning tall but yet white. The other 3 is the recessive yet dominant. In other words, the short and purple. And the 1 would be short and white. That would be down here if you're looking at this, short and white. And so this 9-3-3-1 phenotypic ratio in a dihybrid cross kept popping up in Gregor's experiments. And so, and you're like, well, what, big deal. Well, it led him to believe that not only do the factors, as he called them, segregate during pollen formation, is it led him to his second law, which is actually kind of a controversial law, and I'll come back to what's controversial about it in another video. But it led him to his second law, which is the law of segregation. Not only the law of segregation, but the law of independent assortment. He thought that the factors not only segregated into pollen, but they segregated independently. So they moved independently of one another. And so here's an example of one of the experiments that he did. So he was looking at these pea plants, and what he was looking at here is there's 2 traits in play here: round and yellow. So these are the characteristics of seeds. Round happens to be dominant, and yellow happens to be dominant. Would you believe? Green, which is, I know most of us have seen green peas, but it happens to be recessive, and wrinkled, which is recessive. So if you can look at this and you cross these together, what's interesting is he crossed pure yellow and round with pure green and wrinkled. And what do you get from this particular cross? Well, let's look. What you would get from this is, so this right here is the diploid of this. So the pollen that comes from this plant, the only kind of pollen that would come from this plant is yellow and round. Do you see how I'm separating the homologous pairs? So this is a haploid pollen. I'm not going through all the stages of meiosis. And see this right here? This is the green, which is little y, green and wrinkled, which is little r. This is going to produce green and wrinkled pollen only. So if this pollen and this egg come together, every plant, this is the parental cross, every plant would be, in the next generation, can you do this? Yellow and round. Every plant from this cross would be yellow and round in the first generation. And every plant would be dihybrid in the first generation. And you're like, well, I don't know. This seems kind of basic. You cross this with this and they're all yellow and round. What's the big deal? Well, it's what comes next is kind of fascinating. Like if the F1, they're all yellow and round, but then the F1, if you see, this is weird. If you cross yellow and round and self-pollinate them, yellow and round hybrids, you get 9331 in the next generation because that plant, and I'll just work it out for you, that plant, that yellow round plant produces 4 different kinds of pollen. And so how that happens is that, I hope you're following this. I'm not drawing out the chromosomes, but this is a diploid right here because there's homologous pairs. They're heterozygous for 2 traits. We call this dihybrid. When this segregates during meiosis, you get big Y going into one gamete. Obviously no little y because they segregate. So you get big Y, big R. You can get big Y, little r, big R, and little y, little r. So you get 4 gametes from this plant. And when you get 4 gametes from the plant, and obviously there's 4 male gametes and 4 female gametes, and so when you do the Punnett square, there's 16 possibilities, 9 of which show round yellow, 3 of which show yellow wrinkled, and then green round. So you get this 9-3-3-1 ratio, and that kept coming up, 9-3-3-1. And so Mendel noted it. I'm not sure he understood exactly what was happening, but he was certainly curious. Now this applies to anything. It could be tall and purple. This is the example that I was going over earlier. This 9-3-3-1 is the classic phenotypic ratio that you get in a dihybrid cross. And so Mendel thought that each character was inherited independently. And that's true. I'll just state this. I hope you would understand it, but I'll come with it later. This is true when the genes are on separate homologous pairs, because when they're on separate homologous pairs, they assort during meiosis this way. But if the genes are on the same chromosome, this is not what you get. And it's called linkage. And so here's another example of it. And so in general, Mendel called this his law of independent assortment. And so he was able to detect this. And so he came forth with these 2 basic principles. Law of segregation, meaning that the factors segregate during pollen formation. And if you're considering 2 traits at the same time, the traits segregate independently, and that was his law of independent assortment. And so I hope that was informative, and we will continue the discussion in the future, and I hope things will clear up if they're not clear already. Thanks for watching.